We are happy to welcome you at the online lecture series from the NGS CN Summer School 2024. As most of you already know, NGS CN stands for the Next Generation Sequencing Competence Network. Our network has started as a DFG-funded national collaborative initiative in 2019, and up to 2023, there have been several annual calls for projects to be sequenced at our core genomic facilities across Germany. From 2024 on, you have a new exciting opportunity to co-apply with one of our genomic facilities for a high-cost sequencing project, which can cost up to 1 million euros. If you ask me, it's worth to check it out. Go for it. We leave the link in the footnotes, and here is a QR code for your fast access. Welcome aboard, find a comfortable seat, get a drink, and let's listen to our next speaker, shall we?
Thank you for the kind introduction. I will talk now a little bit more on the application, not on the technique directly, because I think you had in the morning a great overview from Max already to long-read and short-read methods. So I want to talk about medical long-read genomics, long-read sequencing, and it's a little bit connected to the longer consortium we have in Germany or also now more than Germany, to see if we can bring long-read sequencing into clinics for diagnostics and research. And you can see the longer consortium consists of 4 core sequencing centers in Tübingen, Hanover, Berlin, and Aachen. And we have also other groups joined over time and now participate in the experience exchange, exchanging protocols, and so on. So we are getting some support by ONT for consumables, and of course, we have some aims to showing the diagnostic value, using long-read sequencing, and showing that it can be used in routine. We're benchmarking with short-read data, and we're providing for interested people also some SOPs and bioinformatic pipelines to really have a smooth implementation if you want to have short-read sequencing in your site. But this is enough too longer. So the question is, why do we need long-read sequencing? So maybe you know short-read sequencing can sequence whole genomes, and even the first complete genome was built by long-read sequencing techniques. It's broadly available, it's cheap, it's mature, and you have mature bioinformatics. So the question is really why we need to have long-read sequencing, and I try to convince you in the next few minutes why it could be suitable to use long-read sequencing, especially in the case of rare disease patients. And I want to start with some arguments. So, of course, we have short-read sequencing available. We can sequence everything, but we couldn't align anything because with the shorter reads, sometimes they map to different positions in the reference genome and have the same alignment scores, so they're fitting to both locations. So we don't know to which location the read belongs, so that we couldn't align it in this case. So, and of course, of all of the techniques so far for the next generation sequencing used amplification. Also, sometimes the library preps are without an amplification. We have amplification on the flow cell or for cluster generation or whatever. And of course, the polymerases are not perfect, so some parts of the DNA are hard to amplify, and therefore we have also some regions which we couldn't access with short-read sequencing. And of course, the first genome was built with short-read sequencing methods, but in between comparing the last short-read genome and the new long-read genome, there are 130 megabases are missing, which was not included, was only accessible now with the long-read techniques. We have over 3,000 genes. Of course, only a few ones of that are protein coding. However, we missed some genes in this assembly, and we all know we have 24 chromosomes. However, in the last short-read genome build, these chromosomes was in nearly 949 pieces. And now with the long-read sequencing, it's exactly the number of the chromosomes. So we have now really a chromosome from telomere to telomere. And of course, for these techniques, there were also some other things used, like bug sequencing for scaffold, Sanger sequencing, and so on and so on. So yes, there are some advantages using long-read sequencing. And of course, this here is true, so it's cheap, broadly available, mature, and so on. However, with the short-read sequencing, you missed these parts here of the genome, but you miss also maybe repeat expansion, structural variations, or methylation, which could have an important information in a case of rare disease patients. And with this, I want to come to the first example, so which is analysis of patients having a spinal muscular atrophy. And this is caused by mutations in the SMN1 gene. And there was some more or less unlucky event during our evolution where this part of the genome was duplicated. So we have a segmental duplication here with exactly the same genes, but it's turned the other way around, but it doesn't matter. So the sequence of this part is pretty the same like this. Also, it occurs some years ago. There are now some mutations differentiating these genes from this, but still the SMN1 gene and the SMN2 gene, they are not so much differences, maybe around 10 to 20 bases in difference on 27 kb. So you can imagine this is maybe hard to sequence for short-read sequencing, but I will explain this on the next slide. What you have to understand is we have two genes, SMN1 and SMN2. And the important one for the disease is SMN1 because during the evolution there was some change, some mutation, exon 7 here from SMN1 to SMN2. And this is unfortunately in the beginning of the exon, and this part is important for the splicing. So for the SMN2 gene with the T here, we have a lot of transcripts losing this exon 7 and therefore unfunctional protein. And on the SMN1, we have mainly functional protein. So mutations in SMN1 are important and SMN2 are less important because you don't expect a lot of functional protein. So when you lose the function of SMN2, it doesn't matter. However, if you lose the function of SMN1, you lose this functional protein here, which is important for new development and so on. So in patients from having spinal muscular atrophy or showing muscle weakness, poor muscle tone. So they're also called floppy infants because they have difficulties to sit, to stand, to walk. And of course, you can do some analysis by Sanger sequencing or short-read genome sequencing. And in this patient, for example, we could find two mutations. Both are pathogenic. But the question is, if you find both mutations, are they on one allele? So only one copy of SMN1 is affected, or are they on both alleles, so both are affected? Because if both are affected, SMA is a recessive disorder, meaning that both meaning that you can develop the disease. If it's on one allele, it should be another big problem. And when we're now looking at the short-read data, you can see here short-read whole genome sequencing. I'm not sure if you so far introduced in the IGV view, but these small gray boxes are the single reads. So every small gray box is a read which is generated by short-read sequencing. And here in the upper part of the plot, you can see the coverage. So how many times is this region covered by a read? And it looks a little bit like we make a selection for certain parts of the genes, but this isn't the case. When we are looking with a different view, we can see, okay, no, all over the genes are all over the gene are there reads, but here these ones are white and these ones are gray. And white mean they're mapping to this position, but also to another position with the same mapping score. And in this case, they're mapping, of course, to SMN2. So for these areas, which are not covered here in the upper part, you couldn't align a read specifically to SMN1 and SMN2, and therefore you couldn't see if a variant belongs to SMN1 or SMN2. So essentially, it's hard with these two variants found by short-read sequencing or by Sanger to know if they are on the of both two alleles of SMN1 or one, or even if they are on SMN1 or SMN2. So it's really hard to make a diagnostic with this. However, using long-read sequencing, which much longer reads, this makes it possible to really align this specifically to the different genes. And I want to show you this on the next slide. So this is the same locus. So we have again SMN1, like here, However, we use long-read sequencing. You can see here now we have all of the genes reads mapping to this region. We can also find, you have to trust me, these colorful lines are here the mutations. We can also find the two mutations, and we could even see with haplotyping that these mutations are on the two different alleles, so meaning both alleles are affected, and this explaining in the end, because they are pathogenic mutations, this explain the phenotype of the patient we see. And what we can also see, we can see also reads on SMN2, so we can also differentiate if the mutations are on SMN1 or SMN2. And in this case, we couldn't see the mutations, for example, at this spot. We couldn't see this in SMN2, so we know they are SMN1 and they are on two alleles. So we could find a diagnosis for the patient. And this is, of course, important because since a few years, there's the possibility to having a gene therapy or a therapy for patients with SMA. However, it has to be started or should be started pretty early, so the patients getting some gene replacement. So if they lose both copies of SMN1, they get within ribose an additional copy of SMN1 to then have the possibility to have a functional copy and therefore a normal development compared to the patient with joint gated therapy. And therefore, it's important to have this precise analysis, because this therapy is essentially it's very expensive, so around 2 million euros per patient. So yeah. So this was the first example where this long reads could be beneficial. I have also another example for repeat expansions. So you may know in the genome we have some diseases which are caused by the elongation of repetitive motifs. So I think one famous one is the Huntington disease, which is caused by an repeat expansion, but there are also other ones which could be in non-coding regions like the Fragile X syndrome. And of course, these repeats, so the repeat of different base motifs could be in different parts of the gene. They could be in the promoter or on the upstream or downstream regulated regions of the RNA. They could be in the exons or they could be in the introns. And depending where they are, of course, they have different mechanisms how they cause disease. So most of them you can see are repeats of 3 bases, but there are also even longer ones. So I think the longest one, maybe there are longer ones, but the longest one I know is around 12 base pairs. And so and you can maybe imagine so having 3 times 200, so I have 600 base pairs. It's hard to analyze this with short read sequencing where I have only a read length about 150 to 300 base pairs. And this is also in case for the repeat analysis for the long read sequencing could be beneficial to use it. And I have again here an example from our diagnostics. So we have a patient with sensory polyneuropathy and chronic cough. And this is known to be caused by the so-called canvas syndrome. And there is a repeat in the intron 1 of the RC1 gene. And there's one special thing. It's not only the size of the repeat. So for patients which having this disease, it should be more than 300 repeat units. And it's also the sequence of the repeat. So there could be healthy patients having this 3A2G or 4AG motif. However, if I have the 2A3G motif, this could be pathogenic when it's longer than the 300 repeats units. And it's recessive. So also here, haplotyping is important to have information. Okay, how long is the repeat on the one allele and how long is the repeat on the other allele? So we did this analysis with short read sequencing. And of course, there is some bioinformatic tricks to estimate the size of repeat length. And here we get 70 repeats. It's not haplotype resolved in this case. And from the 70 repeats, you would think, okay, no, it's not above the threshold. And I want to shortly explain how these repeat size estimation works. So of course, you're having these short reads. Mostly we use paired end sequencing, so with one and the other read. And if the repeat very short, then it's easily because the repeats span the reads span the whole repeat. So I can just count how many repeat motifs are inside, and then I know how the repeat length. It could be also that the repeats only covered by a part of the read. So then you can somehow maybe estimate the repeat length, but it will be more difficult. And then you can use also read pairs which are only inside the repeat. And with all this information in the end, you can just make in some estimation how long the repeat could be, using also this reads without a specific mapping in the genome. And then you could come maybe to this number we see here. And you can see also here the results. So again, we have the IGV viewer, we have this gray boxes here, which are the reads. And you can see, okay, they go to this repeat regions, and then you can see here the repeat. And like I explained before, we have the 3A GGG motif. So it's the pathogenic motif, and we can see it's longer than the read length we can get. And with the estimation, it seems to be longer than 70 base pairs. However, when we use long read, we can span the whole loci. And we can also see some reads which are having these colorful parts where you can see the repeat motif. However, we can also see some reads which having here these purple boxes with some numbers inside. And these are estimate or show insertions. So here is an insertion around 270 base pairs, and here around 4,900. And with this, you can really estimate the repeat length. So in the end, we could show for the patient it has a repeat length only one allele from 500 base pairs and on the other one of 900. So more than a 70, which was suggested by the short read sequencing. And then we have the exact length, and this fits to the phenotype. So the patient is have these disease. This I will come to the end of the talk, and I thank you for your attention. And I want to acknowledge some people. So the large team here in Aachen contributes to the analysis of the cases. We had some collaboration partners at the MGZ Munich, and of course the other PIs of the longer project in Tübingen, Berlin, and Hanover. And we are also get some support by ONT and NVIDIA. Okay, and last but not least, if you're interested in long read versus short read as podcast from the NGS CN or the WGGC. And we had a great discussion about long read versus short read. So if you want to dive deeper in this material, so you feel free to listen to this podcast.